Okay. So quick recapitulation what we did in the previous class. So who will tell me what is the difference between a coding strand and a template strand? Which strand is actually responsible for the formation of the RNA? Is it the coding strand or the template strand? Does anyone remember? Absolutely. It's a template strand. Exactly. It's the template strand, right? Coding strand is not responsible for the formation of mRNA. A very important thing to note down. Okay. And then we also spoke about this, see, we're talking about, see, your book is kind of, at certain places, it has explained things about the prokaryotic, in prokaryotes. At certain places, it's explaining with respect to the eukaryotes, because the process is altogether different in prokaryotes and the eukaryotes. When you compare prokaryotic transcription and eukaryotic transcription, it's altogether different, because the RNA polymerase, for example, in prokaryotes, it's different. We have RNA pol 1, 2, and 3 there. Okay. But in eukaryotes, okay, we have different subunits of, RNA polymerase. Okay. We call them alpha, epsilon, gamma, theta, beta, right? So, but it kind of, we have to play around in this chapter, because at certain places, for example, when it talks about here in RNA polymerase, so we don't have RNA polymerase in eukaryotes. Okay, we have some, as I said, that we have something called an alpha, beta, gamma, epsilon, right? These are the various units of eukaryotic RNA polymerase. So here, this is an RNA polymerase with respect to the prokaryotes. Okay, but then what happens? It goes back and explains here, the eukaryotic gene. Okay, so there is actually a lot of difference between a prokaryotic transcription and translation and the eukaryotic. But let's go with the book. Okay, so for the eukaryotic gene, if you remember, I said that every eukaryotic gene has two parts: a structural gene and the regulatory gene. Right? This is a recapitulation of what we're doing right now. Structural gene, we said that if this is a structural gene, it's going to be transcribed into the mRNA or RNA. So this gene can be full of introns as well as the exons, but we need to remove the introns and have only exons at the end. Okay, so that is a structural part of it. But then we also have another DNA sequence, okay, which is called as a regulatory gene. Okay, so regulatory gene is which basically controls the transcription, the way driver controls a car, right? Similarly, the regulatory gene controls the transcription. Okay, so is your regulatory gene transcribed? No, regulatory gene cannot be converted into mRNA. Only structural gene is converted into mRNA. Remember it forever. Okay, it is not transcribed, the regulatory gene. Okay, so it is basically just there because it will control the transcription. Okay, so what is present in this regulatory gene? In this regulatory gene, we have the promoters. Now your question might be, what is a promoter? I explained you last time, promoter is simply a DNA sequence. For example, as you can see here, this is thymine, adenine, thymine, adenine, adenine, adenine. Okay, this is a promoter. Okay, and this has been called as a Tata box. Okay, rather than saying thymine, adenine, thymine, adenine, adenine, the shortest name, it has been called as a Tata box. Okay, so Tata box, what is a Tata box? It is a promoter, a DNA sequence, okay, where actually the RNA polymerase binds. Okay, so this promoter, okay, identifies the start point of the transcription. Okay, so it is very, very important, and in a while we'll talk about how RNA polymerase will need the help of some other proteins called as the transcription factors to bind to this promoter. Okay, did you all understand what is a promoter? What is a promoter? Promoter is a DNA sequence or a specific sequence. Where is it present? It's present on the DNA. Okay, so what is so special about the promoter? Promoter is a place where the RNA polymerase will bind. Is it clear? It is identified as a start point. Okay, is it clear to everyone? So for example, if my transcription is going to begin from here, okay, as you can see, this is a start of transcription. Okay, so if the transcription, this is a start point, Okay, and then there will be a definitely a termination point as well, the stop signal as well, right? So this starter box, or you can say the promoter, is little far away from the start of the transcription. Okay, usually maybe 20 or 25, as you can see, 25 base pairs, right? It's 25 base pairs away from the start of the transcription. Are we okay with everybody? Did you all understand what is the role of, very important, what is a promoter? The question says, what is a promoter? What is the role of promoter? Very, very important question, right? So as you can see here, this is a promoter over here. There's a Tata box over here. Okay, so we also have many other promoters, like we have a cat box, we have the GC box, but we'll go with your book. Okay, in the book it says only the Tata promoter. Okay, where you have thymine, adenine, something like that. Okay, and we'll talk about this later on, how it's so this is the definition which I've put here for for the promoters. You can just read it out. Okay, and after that, okay, now once the transcription is started, okay, once the transcription is started, what is very important is that you have to understand that in DNA transcription, okay, if you remember that in DNA transcription, we spoke about so many enzymes, the helicases and the topoisomerases. No, we don't have all those enzymes over here. We'll talk about those factors as well, Muhammad. Okay. We're not going, as I said, that it's kind of, we're not going into very detailed manner, in a detailed way in the transcription and translation. But yes, it is not very detailed, but yes, it is detailed. We're going to talk about that in a while. Okay. We didn't finish the promoters yet. Okay. We're going to talk about the transcription factors. Okay. Which is coming up. Okay, first of all, let's simply try to understand a general thing. Okay, general transcription. In the general transcription, we last time said that the transcription takes place in the nucleus, right? We said that last time the RNA polymerase is the enzyme which does not need primer. We discussed all that, right, last time. Okay, because it's the RNA polymerase which will form the mRNA. Okay, so what happens generally, if we talk about RNA polymerase, binds to a specific DNA sequence called as a promoter. Okay, but in a while I'll explain you what are the other factors which are required in the eukaryotic transcription. So I'm just telling you generally what happens. So if the if the RNA polymerase binds to to the promoter region, okay, that means transcription will start from here. Okay, as a transcription will start here. Okay, so DNA unwinding and everything is done by the RNA polymerase itself. We do not have helicases over here. So DNA unwinding and winding it back again is taken care by the RNA polymerase itself. Now the direction, very important. Same as DNA polymerase. RNA polymerase can elongate this mRNA chain only in 5' to 3' direction. It's the same thing. It will read the sequence in the 3 to 5 direction, right? But will form the mRNA in the 5 to 3. So reading in 3 to 5, but formation of the new strand of mRNA will always take place in the 5 to 3' direction. Is it clear? Everybody till now? So we don't have DNA unwinding enzymes, helicases. The true and false will come, based on that. So you have to say that no, DNA unwinding and or rewinding it back again is taken care by the RNA polymerase itself. So this is called the transcription bubble. As you can see here, it resembles like, the replication bubble, right? So this is a transcription bubble where one strand of the DNA, which is called as a template strand, will be used to form the mRNA with the help of an enzyme called as an RNA polymerase. Okay, so RNA polymerase 2 will be there for the mRNA synthesis. Okay, and then what happens? How will all this stop? How will your transcription stop? Your transcription stop once you have a termination signal. If you remember, I told you that on the DNA, give me a minute. If you remember, what's wrong? If you remember, I told you that we have something called as a stop signal over here. Okay, this stop signal over here. So once a stop signal will be there, we have a start signal and we have a stop signal here. Once this sequence will be encountered by the RNA polymerase, so that gives it the signal that, okay, you need to stop here, we are done. We don't need to make more mRNA or the RNA. Okay, so termination of the RNA chain will occur only when the RNA polymerase will bind to this sequence. Okay, so that is like a general thing you need to know about the transcription. Okay, so is it okay till now? Any question? And if you can see here, there's no primer over here. We do not need any primer for the RNA polymerase. No, of course not. There's no primer over here. Okay. And then, so before we talk about, what happens in the post-transcriptional modification, okay, let's talk about the transcriptional factors. Even though they have not mentioned here transcriptional factors, but there is, at the end of this, this presentation, there's a slide about the transcriptional factors, okay. So what are the transcriptional factors, okay? So basically what happens is that eukaryotic, please pay attention everyone, eukaryotic RNA polymerase, we just said that promoter is that area. If this is my DNA, which has to make the mRNA, we said that promoter is that data box, for example, is that area on the DNA where your where your RNA polymerase will, which will identified by the RNA polymerase. But the problem with the eukaryotic RNA polymerase is that the eukaryotic RNA polymerase cannot identify this promoter sequence. Okay, so it is a little different in eukaryotes. It's a little complicated in eukaryotes. So for example, even if RNA polymerase is around, it cannot actually, can say that, it's blind towards the promoter. It cannot identify the promoter. So it needs the helping proteins, helping factors. Remember that transcription factors are only there for the eukaryotic transcription. The prokaryotic RNA does not need transcription factors. Okay. So what happens? So before the RNA polymerase can actually bind to the DNA to start the transcription, what is happening here that the transcription factors bind to the promoters first. That means if this is, what's the problem with it? Okay, that means if this is the promoter area, okay, this area is first of all identified by the transcription factors, because I just now told you that RNA polymerase has very less affinity for the promoter area. You can in simple language think that it's a blind, okay. RNA polymerase is blind and it needs the helper, and that helper is your transcription factor. Is it clear? We also call transcription factors as DNA binding proteins because they bind to the DNA. Is it clear about the introduction? Is clear about the transcription factors? Everyone, did you all understand? Okay, but now, so RNA polymerase enzyme will be recruited by these. Yes, that is the next step which I'm going to talk about. So now we spoke about that. Yes, Mohammed, you're right. So now we spoke about that RNA polymerase cannot identify the promoter. It needs the help of the transcription factors, right? So if you see here in the next diagram, okay, I don't know what's wrong with I'm not able to flip the slides. Okay. If you see here in the next diagram, look at this. Okay. This was the promoter area. Okay. So you will have many because, you don't have to bother about what is this TFA, B, C, D, because we have plenty of transcription factors and nobody will expect to remember their name at all. Of course not, right? We usually write them as TF. So TF1, 2, 3, 4, A, B, C, D, no need to worry about that, right? So first of all, we have the transcription binding protein, which will bind to the promoter, right? And then, when one transcription factor will bind, all others will follow, like all of them will follow. So once you have this transcription factors binded, or bound to the, the promoter, this starter box is a promoter, then these transcription factors will attract your RNA polymerase. Okay, so then it will attract your RNA polymerase, and then our RNA polymerase will come and the transcription of gene will be started. Is it clear? So we don't have to know a lot of things about the transcription factors. All you need to know is that they're very much required for the transcription of the eukaryotes, because your RNA polymerase cannot bind unless and until all the transcription factors have bound, and then only they recruit or they attract the RNA polymerase. Or you can in other ways say that transcription factors are like in charge of the transcription. I will not go into details. Even if you don't want transcription to take place, you want to stop the transcription, okay, you want to block RNA polymerase, so it is definitely in the hands of these transcription factors. So you can say that these transcription factors are so very important. They regulate the gene expression, turning it on, turning it off. Everything is within the hands of these transcription factors. Okay, so the starter box is initially, recognized by these DNA binding proteins. Okay, we call it a Tata binding protein. You don't have to know all that, anyways. Okay, are we okay till now? Did you all understand the role of, very important, did you all understand the role of the transcription factors? Okay, so you can simply say that your RNA polymerase cannot position itself, okay, unless and until it sees the transcription factors there. Okay, so then only it can position itself and then start unwinding, and then the process is same, the elongation, everything is similar, right? It will start, because whether it's an RNA replication, whether it's a DNA replication, whether it is a transcription or whether it's a translation, you will always have initiation, elongation, and the termination. Okay, so then after that the RNA chain will be elongated, and it will continue to elongate unless and until the termination signal will not come, which tells the RNA polymerase that you have to stop now. Okay, no need to start forming, no need to continue forming the mRNA or the RNA. Okay, so there's one topic which is at the end of this chapter, okay, the transcriptional control. I'm going to explain that along with this here, so that then later on I may not need to explain it here, because we've already started the discussion about transcription. Okay, so any questions till now? Are we okay till now? Okay, then also there's something else which is mentioned in your book is about, even though it's not in detail, okay, it's just a superficial thing. What are enhancers? Okay, so other than having these, transcription factors, we have something called as the enhancers or silencers or response elements. Okay, so the question says that what are silencers? What are enhancers? As the name itself says that enhancers are basically what happened. They are another group of transcription factors. Okay, they enhance, they speed up the transcription. Okay, by binding to certain DNA sequencings. That means, oh, what's wrong with it? Okay, that means, you know on these, on the same DNA, you will have certain sequences, okay, to which the other transcription factors will also bind, and they will enhance the transcription, they will increase the rate of transcription, so that mRNA synthesis can quickly take place. Okay, so they're called as the enhancers. That is all you need to know. Okay, so you might be having you might be having a specific sequence, okay, which can enhance the transcription. Okay, and then we'll not go into detail. Then we have an activator protein which can bind to it, and I will not explain you all that to confuse you further. No. And then the silencers or repressors. Okay, so as the name itself says that we also have some sequences, okay, which can actually slow down the transcription. Okay, and those proteins are called as the silencers. And once these silencers bind to the DNA sequence, they can inhibit the start of the transcription, or they can simply stop the transcription. Khalas, finish. Okay, we don't want transcription to take place, right? That is all part of the gene regulation. Okay, how the genes are being regulated, that depends upon, that varies from one cell to the other cell. Is it clear about the enhancers and the silencers? Okay, and then there's something also called as a response elements. Okay, so what happens, response elements are also basically, for example, if this is my, G, for example, GC, for example, this is a sequence on my DNA, GC, right? So there might be a sequence maybe on the, there might be a sequence maybe on the mRNA, okay, a short sequence which is called as a response elements, okay. So that is present within the promoter, right? For example, this whole thing is a promoter. So we have a small sequence which is called as a response element. So what is the function of this response element, right? This response element can also bind to certain transcription factors and regulate the transcription of the genes. So I will repeat once again. Let's say, for example, this is my Tata box, or you can say this is my promoter. Okay. So within this promoter, I might be having a short sequence, like maybe like adenine, thymine, guanine, cytosine, a short sequence here maybe, okay, which is called as the response element. It is present within the promoter. Okay, so what happens, what is the function of this response element? So we have certain transcription factors which can also bind to these response elements and then regulate the transcription of genes. Any questions till now? Are we okay till now, everyone? Any questions about transcription factors, about the process of transcription till now? Okay, and then we just have one more topic, we'll talk about that, the zinc fingers and all. We'll talk about that. It's just a 2-minute topic, and we'll talk about that at the end, how they bind to the, how they have the affinity towards the DNA. Okay. Now we have to remember that one of basic difference between the prokaryotic and the eukaryotic transcription is that there is no post-transcriptional modification in the prokaryotic DNA. I mean, in the prokaryotic DNA, once it's formed and it's formed, khalas. First of all, you know that it's not formed in the nucleus. Okay, it is not from the nucleus. The prokaryotic DNA is formed in the cytoplasm. However, for the eukaryotes, the transcription takes place within the nucleus. So, what is a post-transcriptional modification? As the name itself says, that post means after, transcriptional modification. And if you remember, we spoke about it. The first thing is, once the template strand, okay, once it forms the mRNA, this is a mRNA, but this is like an immature mRNA. You cannot send this mRNA to the cytoplasm because it will be degraded in the cytoplasm. It will be, attacked by so many enzymes, okay, like endonucleases, and it will be degraded. So we cannot afford to send this to the cytoplasm. So what do we do here? Okay, we do something called as a poly A tailing and the capping, as you can see here. Okay, remember now, the question says that from which end of the pre-mRNA the capping is done. Okay, so actually both the ends are capped, right? But now if you talk about, what happens in the 5' end, let's say this is a 5' end, and let's say this is a 3' end. Okay, in the 5' end, yes, in the 5' end, what is basically, what do we do? Okay, there is something called as, methyl guanine, okay, is added on the 5'. I will not go into detail what are the chemical formations here. We don't have to know all that. You simply have to know that the 7-methyl guanosine is added here, okay, is added on the 5' end. Okay, I think we had a diagram also for that. There's something wrong with my presentation today. Okay, look at this. This is the 5 dash capping done over here. Okay, so simply you have a kind of untranslated part. We call it a 7 methyl guanosine. Okay, so methylated guanosine is there, and you know that methylated guanosine will not be translated. So it's something like, just to put a cap. If you remember, I gave you an example last time, the way your shoelaces, they have that plastic caps, so that the ends of the shoelaces are not spoiled, isn't it? So similar kind of, we can say the capping is something like that. So now they are secure. Now they are, kind of, they cannot be attacked by any other enzyme here when it reaches to the cytoplasm. Okay, and you will see that. So that's the basic reason, why they are capped here, or poly, the tail is added over here. But then capping also will help in the, 5 dash capping will also help the ribosome, to bind it during the translation. Okay, so for example, when I will start translating the mRNA into protein, which is the next topic coming up, right? So if this mRNA is not capped, I mean, I don't have this 5 dash, 7 methyl guanosine over, it's not capped, right? For example, it has to be capped here. Let's say this one is not capped, right? So the uncapped mRNA cannot bind to the ribosome for protein synthesis. Did you all understand? So capping is, this is the like second important thing why capping is excuse me. This is the second important thing why capping is being done over here. Is it clear till now? Okay, so so that was about the 5 dash end. So similarly, the other end is also kind of sealed. If you see here, this end is also sealed here by an untranslated position. What is that untranslated position? So almost adenine is added here, like adenine, 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 a long tail, as you can see over here. Look at this, called as a poly A tail. Okay, so adenine is added on the other end. Okay, so adenine is added at the excuse me, at the 3 dash end. You have to remember these points, right? And this adenine, maybe sometimes, 250 base pairs of adenines will be added. Adenine will be added, like, adenine, 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 250 times it will be added. Okay, and this tailing is again very important, because this tailing is going to protect the mRNA from degradation. That's one thing, right? Both capping, capping on both the ends, as I told you, that it basically protects the mRNA from degradation, but it will also help the export of mRNA to the cytoplasm. Okay, so transportation, okay, sending it to the to the cytoplasm will become easier if, see, this RNA is not capped. Neither I have a 5 dash, 7 dash methyl guanosine here, neither I have a poly A tail here. So this can never be sent to the cytoplasm, never ever, right? It will be exported only if it has this post-transcriptional modification. Now it is, look at this, it has a cap on this side, it has a tail on this side, and now this RNA can be exported, and it can also start with the, it is ready to begin the translation in the cytoplasm. Are we okay about it? This was the post-transcriptional modification. So what is the other thing which is to be done over here? I think we discussed that in previous chapter. Does anyone remember? There's something else which we need to do here again, other than, capping and polyadenylation, adding the tail. There should be one more thing which we need to do before we send mRNA to the
Splicing.
Absolutely. Removing the introns, splicing, absolutely, right? Remember the SNRPs, the spliceosomes, the snRNA, small nuclear RNA plus the protein, right? They are very, very important in removal of the introns, right? So as that will be done, and now it's ready to be exported in the cytoplasm. So with that, we finished one part of the chapter, which is the transcription. Okay. Do we have any questions till now about the transcription? Then I'll move to the translation. Are we all okay, guys? Any questions about transcription? No, it's clear. Okay, Alhamdulillah, good. So before we start translation, who will explain you what exactly is translation? What do you mean by translation? Yes, you can switch on your mic if you want to. What is it? Translating mRNA to the amino acid. Yes. Protein synthesis. Very good. Okay, where do we need to go now? Making a protein. Like now we have the recipe, right? We have the recipe of making a cake. We're going to make a cake, right? So we have the recipe of making the cake, right? And that recipe, we got it from the mRNA. So mRNA knows what, for example, if you try to cook something in the kitchen, you first of all download a recipe, right? You keep your phone in front of you. That's what I do if I'm trying something new, right? So you keep a phone in front of you and try to copy the recipe, right? So mRNA is something like, a cake making recipe, or you can say protein making recipe over here. Okay. And translation is something, okay, you take that recipe, you go to the kitchen and you prepare, whatever. If you want to make a cake, you'll go ahead and start your cake making, right? So translation is something, we're going to make a protein now. Okay. For cake making, you'll go to the kitchen. Okay. But for translation, for protein making, where should we go? We should go to the cytoplasm. But where in the cytoplasm? We should go to the ribosomes. Absolutely, guys. Right? We should go to the ribosomes in the cytoplasm. So, like the way kitchen is there for your cake making, we have ribosomes here for the protein synthesis, for protein making. So I have a recipe, which is an mRNA. I have the ribosomes, which is like a kitchen for me, where I'm going to make the cake. And then I need, kind of maybe a recipe. Just imagine the recipe is, for example, in some Chinese language. Okay. So what would you do? How will you make your cake if the recipe is in the Chinese language? What will you do? Yes, ladies, boys? You might need a translator. Exactly. Now, all our phones have a Google translators, right? What would you do? Okay. So, for example, if that's what I do, if I have a message in Arabic, I translate it in English, right? Or if we have English, we translate it, in Arabic. Exactly. So the translator who's going to translate your, recipe on the mRNA, okay, which was in which is in Chinese language to Arabic or English, the translator is your tRNA. So you cannot make a cake unless you have a kitchen. You cannot make a cake unless you have a, the recipe, okay, which is an mRNA. You cannot make a cake unless and until you have a translator, because you cannot read Chinese language. So same thing, we cannot make a protein unless and until we have a recipe for protein making that is within the mRNA. We cannot make a protein unless and until we have a ribosome where this protein will be made. Okay. We cannot make the protein unless and until you have someone who can actually convert this information, who can act as a translator, convert the information on the mRNA into the information which can be read by the ribosome, right? And that is your tRNA. I tried to make it as simple as possible. I hope I did. I was, I succeeded in doing that. Did you all understand? So what do we need? What are the requirements for the protein synthesis, right? The requirements are we need an mRNA, which has a sequence for protein making. We have the ribosomal RNA, which is there to make the ribosomes, because if you remember, we said that ribosomal RNA and ribosomal proteins, they make the ribosomes. And then we have the tRNA, of course, right? To bring everybody close to each other, because tRNA, okay, if we go back to the tRNA, I would like to show the structure of tRNA. So that's how tRNA would look like. Okay, tRNA is, this L-shaped molecule, and its main function is, what is the function of tRNA? The main function of tRNA is, as per the codon present on the mRNA, okay, it will basically bring that amino acids. We have 20 amino acids in the cytoplasm, just 20 amino acids. Okay, but we might have more tRNAs, right? We might have 45 tRNAs, maybe 50 tRNAs, right? So that means we have more than one tRNA for a particular protein, because we just have one, we just have 20 amino acids. So what is the main job of tRNA? The main job of tRNA is to bring the amino acids, okay, to the ribosomes, okay, where they are decoded, okay, where the information present on the mRNA, okay, which will be decoded into the long amino acid chain, isn't it? So that building will take place only if your tRNA will carry the amino acid. That is so very important, right? It's very important. In a while, we'll talk about the charging of tRNA, okay? Soon by the enzyme amino acid tRNA synthetase, okay? And remember that it's the 3' site on the tRNA to which the amino acid binds. Sometimes the question says that name the side or name the sequence, or not the sequence, name the side, okay, to which side of the mRNA, tRNA, will the amino acid be attached? It will be attached to the 3' side of the of the tRNA. So this is an amino acid attachment site, not the 5', it's the 3' one over here. Okay. Okay, now another question. So how many amino acids can one tRNA bring? How many amino acids can one tRNA bring? It can bring only, yes, it can bring only one amino acid. Based on, see, that is very, very important. Sometimes the question says that, where do we find the anticodon? Remember, one arm of tRNA can bind to the amino acid, okay? The other arm of this tRNA, as you can see here, this is an anticodon arm, right? This is an anticodon arm. This will be totally opposite, complementary to what we have in the mRNA. Remember, 3 bases together on the mRNA, it's called as a codon. For example, you have CUU. It is a codon on the mRNA. So codon is always on the mRNA, whereas anticodon is always on the tRNA. And these two, codon and the anticodon, right? These triplet bases, they're complementary to each other. Okay, and that is very, very important, because when it comes to alignment of the tRNA with the mRNA, right, they bind to each other because they're complementary to each other, right? So that is very, very important. So based on what codon we have, okay, that tRNA, that charged tRNA will be picked from the cytoplasm. Okay, in a while we'll talk about the genetic code. Okay, so if, for example, the CUU codes for the alanine, for example, it codes for, just in case, it codes for alanine. Okay, so the tRNA which has an alanine over here, only that one will be brought over here. Okay, if the CUU codes for the leucine, so only that tRNA will be brought here, which on the other side, it has a leucine here. So that's why I said that tRNA plays a very, very important role here. Is it clear till now? Any questions till now? So what is if the question says, what is the codon recognition site on the tRNA? What will you say? Name the codon recognition site on tRNA. What is it? Codon recognition site on tRNA. What is it? The site which recognizes codon on tRNA. What is that called? It's called an anticodon. Absolutely. We call it the anticodon arm, okay, or just the anticodon. That's the recognition sequence for the codon. It recognizes the codon on the mRNA. That is very, very important to remember here. Okay, so that was about the messenger RNA as well as the tRNA. Okay, so the enzyme, we'll talk about in a while about the activation of amino acid. The enzyme bonds the activated amino acid to 3' terminal, okay, of the tRNA by an ester bond, right? So this is the 3' terminal because it provides the free OH group, right, over here, okay, in the sugar over, the pentose sugar over here. And leucine or any amino acid will bind to this over here, okay, and the bond here is the ester bond, okay. So that was all about. And remember, each tRNA is specific for only one amino acid. As I told you that we have, we have 20 amino acids. For 20 amino acids, we definitely have 20 tRNAs and 20 enzymes also. I'll tell you in a while. But then we don't have only 20 tRNAs. We have more than 20. We might have 45 to 50 tRNAs, right? That means for one amino acid, we have more than one tRNA. But that tRNA is specific for that alanine or a leucine, right? Whatever amino acid it is. Okay, and now the joining reaction. Okay, I just told you that amino acid binds to the tRNA at the 3' end, okay, of the tRNA, 3' terminal of the tRNA. But how is this reaction catalyzed? Okay, which enzyme is going to catalyze this reaction? We have an enzyme called as an amino acyl tRNA synthetase. Okay, so that is the enzyme which catalyzes binding of amino acid to the 3' terminal of the tRNA. Okay, so this is the enzyme amino acyl tRNA synthetase. Okay, and as I told you right now, as I told you initially, that there are 20 different enzymes. So every amino acyl tRNA synthetase is, specific for that amino acid. Okay, so we have 20 amino acids. We might have 45 to 50 tRNAs, but we have also 20 different enzymes, amino acyl tRNA synthetases for the specific, amino acid which binds to the corresponding tRNA. Is it clear? And every enzyme here has a specificity, because this is a very, very crucial step. This step is a very, very crucial step, very important step. If there's any mistake over here, that means, the protein synthesis is not going to be the normal. It's very, very important. So that is why every amino acyl tRNA synthetase is specific for that amino acid in the tRNA. Because, for example, if we had a different codon here and we got a different amino acid over here, that means then there will be tafra, mutation in the DNA. So this is a very, very important step, right? Which the enzyme has to take care of it, so that the right amino acid is bound to the right place on the tRNA. And we call this as an activation of amino acid. We'll talk about that once again in a while, once we start translation. Are we okay till now? So we'll come back to this in a while. Okay. So now let's talk about, before we start the process of translation, let's talk about genetic code. So before I can explain you genetic code, can you tell me what is the genetic code? Does anyone know what is a genetic code? Or have you ever studied this before? What is a genetic? Have you ever studied this before? What is a genetic code? And what exactly does it signify? What do you mean by genetic code? Why is it called the code? A to T? No. Anybody else? What do you mean by genetic code?
The basis.
Basis, yes, very good. Basis on what? Very good. No. Who will tell me what is the genetic code? Writing of protein bases on RNA. Okay, I'll take genes coding for what? Absolutely. Not genes, the codons. Okay. Yes, Tanya. What about others? Okay, so the genetic code looks like this. So before I scare you all, remember that this table needs not to be prepared. You don't have to remember this genetic code at all. Nobody's going to ask you what does CUU code for, what does UCA code for, what does CCC. Yes, the only thing which I'm going to ask you is basically maybe about the initiation codon and the stop codons. That is the only thing we might ask you from this table, right? What is an initiation codon? What does it code for? What is a stop codon? Its function, right? So that is the only thing which you might be asked about it. Okay. So let's talk about the genetic code. So genetic code is basically, I don't want to go back into history of it, right? So there was a scientist, his name was Nierenberg, right? He was awarded Nobel Prize in maybe 1960s, okay, because he was the one who basically broke this genetic code and told us that then there's a procedure written over here how he did the experiments. For example, he took poly U, okay, for example, he took uracil, for example, okay. And then what he did is like basically he tried to search, in the genome that, okay. And then he found that, okay, this is the triplet which codes for, phenylalanine, for example, okay. So that's what he did, okay. And for example, if for example we have this code on the mRNA like ACA CAC, right? So what happens, he took an artificial sequence to find out, and then he found that, oh, ACA is the one which codes for this amino acid. So no need to kind of go into these sequences in detail, how they did it, right? Because there is, kind of a lot of other things which they did actually, other than just this. Okay. So what is very important to know? You need to know the features of the genetic code. That's so very important. Okay. How many bases form the code? We just have 4 bases, right? So four: adenine, thymine, guanine, adenine, thymine, guanine, and uracil, right? So these 4 bases, thymine, we don't have it. We have a uracil here, right? So these 4 bases, whether we have a uracil, whether we have an adenine, whether we have a guanine, whether we have a cytosine, right? These 4 bases will have formed the 64 codons. You need to know the numbers as well. So we have altogether 64 codons if you count them all, right? Remember that. So out of these 64 codons, what do we have? We have 61 codons, which are called as the sense codons. Why do I say sense codons? Because only these 61 codons code for a particular amino acid. So 64 minus 61 is how many codons are left now? We have 3 left now, yeah, which code for the 20 amino acids. Absolutely, Mohammed, right? And then we have 3 codons, right? And these 3 codons are like, UAA, UGA, UGA. They're called as the nonsense codons. Why nonsense codons? Because they do not specify for any amino acid. No, of course not, right? So they specify for, we'll talk about that, how the release factor is introduced, okay? So they simply tell the mRNA chain that you have to stop now, no need to form more protein, no need to form a polypeptide chain, stop now. So those are called as stop codons. Is it clear to everyone? So how many codons? 64 codons overall. 61 are the sense codons which code for the amino acid, and then we have 3 codons which do not code for any amino acid, but they are called as nonsense codons because they do not code for anything, but they are just there to stop the translation of the proteins. Okay, and then AUG is a start codon. Okay, so we also count this AUG in the 61. Okay, because sometimes students do get confused. What about AUG? What is AUG? Is it counted in the 61 or these 31? AUG is counted here in the 61 because I know that it starts the protein synthesis, but it also codes for the amino acid called as a methionine, right, in eukaryotes, right? So that is why it is considered as a sense codon. And but what is important to remember here that AUG is the start codon. So you will always find this codon at the beginning of the mRNA. So protein synthesis will always start with the AUG, and methionine in the eukaryotes will be always the first amino acid. Okay, so the first amino acid in the eukaryotes will always be methionine, but then there's a later thing, then we have post-transcriptional modifications. So you might sometimes, you need to remove this methionine, okay, after the protein is translated. But usually methionine is the first amino acid which you will always find at the beginning. Are we okay, everybody? Now, very important point to remember. Okay, we need to know the various characters of the genetic code. What does that mean? Okay, that means, for example, we say that genetic code is composed of triplets, right? So you need to learn all these and you need to understand the meaning of all these. When I say what are the features of the genetic code, that means, what is so special about the genetic code? First of all, genetic code is always made up of triplet codons. You will always have, for example, G, C, G, okay, G, A, or A, right? You will never have 4 bases together. You will never have A, G, U, A. You will never have that, right? Of course not, right? So it is basically the codons are on the mRNA, and each codon is always used as a triplet. Okay, and each triplet will code for a particular amino acid. Is it clear? Any question till now? We are all okay, right? We are not going fast, we are not going slow, we are going okay. Just let me know if you don't understand anything. Okay, now moving on further, we said that genetic code is non-overlapping. What does that mean, non-overlapping? Non-overlapping means, for example, the codons are read one after the other, right? So you don't have, for example, if you see, you have AUG, khalas, it codes for one amino acid. Then you will have, for example, GAU, right? That will code for another, they're not overlapped, okay? And there's no punctuation over here. If you see here, it's non-punctuation. There's no comma or semicolon in between here, okay? So that is called as they are unpunctuated, right? So if you see a sequence like this, right, you don't need to put comma or any punctuation over here because you know that it's AUG will be one codon, GGC will be another codon, CAA will be another codon. So it can be continuous as well, okay? Is it clear? Then genetic code is degenerate. I have seen a lot of people, a lot of students do not understand about what is degeneracy, okay? When we say that genetic code is degenerate, okay, that means that, okay, we know that 61 codons code for the 20 amino acids, right? So that means, see, there are just 20 amino acids, but there are 61 codons, okay? Degenerate means that one codon, degenerate means that one codon, okay, might code for many amino acids. Okay, I'm sorry, one amino acid will be will have many codons. Okay, for example, if you see here, serine, okay, is an amino acid which has 6 codons. Okay, serine has 6 codons, so you can't help that. That's okay. Okay, so as you see here, it's coded by 6 triplets, 6 codons, but that's okay, right? It's degenerate, right? So degeneration means one amino acid has more than 1 codons. That's perfectly okay, right? Or even if you see here, glycine, okay, glycine is also one amino acid which has 4 codons, but that's okay, right? But this is called as what is it called? Degenerate, or degeneracy of the code. Okay, but what is so important, the important thing is, even if I have 6 codons for serine, right, but you will see that the first 2 bases in the codon will always be same. For example, if serine is coded by G G C, okay, G G U G G T, right? Oh, we don't have T. Okay, sorry, C. Let's say once again. Okay, or let's say A. Okay, so look at this. Every time I have first 2 bases similar, but it's only the third base which changes. This is very important. This minimizes the tafra, the mutations. Okay, mutations in our in our genome. Is it clear? What is the meaning of degenerate? This is something which students always, they get confused about. What is it? What do you mean by degenerate? Yeah, only first 2 bases are similar. Yeah, the third base will be different facet. Okay, the genetic code contains start codon and stop codon. We spoke about AUG and the UAA or UGA, right? The stop codons. Okay, and then genetic code is universal. That means we have the same table for everyone, whether it's for a fish or a human being or a bacteria or an elephant, right? It's like a universal, right? The codons are same for the same amino acid in all the species. If, for example, ACA, okay, or you can say CCA codes for the proline, so it's going to code for proline not only in me, it's going to code proline in elephant, in bacteria, in viruses, in fox, in tiger, everywhere it's going to code. So it's universal. Okay, is it clear till now? So these are things which you definitely need to remember, right? So that's why I wrote them here so that you don't forget it. Okay. So these 5 points are very, very important, which you need to know, right? Because as I said, that genetic code is very, very important, right? It codes, as I told you, you don't need to know all this. No, you don't need to know all this. This is like you can say this is like a dictionary of the translation. Okay, for example, if you want to look upon a word, you will open your dictionary, right? Your Oxford dictionary, and look that word in the dictionary. You will not memorize the entire dictionary, right? So this is something like a dictionary of the translation. Okay, whatever you want to look for, you can come here and look. Okay, what is AAU? What does it code for? What is CAC? What does it code for, right? So that's why you don't need to memorize the table over here. Any questions about it now? So which is the initiation codon among all these 64 codons? Which is the initiation codon? AUG. Very good, Tanya. So others, what about which is the first amino acid in a protein always? The first amino acid in the protein will be the first amino acid. So you have to always listen. It's the methionine in eukaryotes, because we'll talk about difference in the prokaryote. Prokaryote we have the F-formyl methionine. We'll talk about that. Okay, but does that mean you will have methionine always at the beginning? You cannot have it in the middle? No. Even though it is used as a start codon, but you can have methionine in the middle as well, right? You can have AUG in the middle as well. But yes, AUG is definitely there a start codon, right? But AUG can code for a methionine anywhere else as well, in the middle of the chain as well. So you don't have to worry about that, right? Are we okay about the genetic code? So now before we start translation, I want all of you to watch a video, okay? Uh, then we can talk about the translation, okay? I'll show you two videos. First, I'll show you the transcription, which we finished, and then I will move on to the translation so that you can, now kind of, join the dots now, and then I'll move to the translation. I hope this time my video will work. Last time, it really gave us a tough time. Okay, I'm just trying to... So I'm just trying to upload a video. Mhm. Just let me know if you cannot hear the sound or watch the video, okay? Just let me know. Switch on your mics and let me know about.
Transcription is the first step in the expression of a gene. It is the formation of a specific RNA sequence from a specific DNA sequence. Transcription initiation occurs at a promoter, a region on the DNA, where RNA polymerase binds. RNA polymerase attaches to the promoter and begins to unwind the DNA. At the initiation site, the polymerase begins reading the DNA template strand and building a complementary RNA strand from free nucleoside triphosphates. The RNA strand grows by the addition of these substrate molecules to its 3' end. This is the elongation phase of transcription. Note that the bases in the nucleotides of the RNA strand form pairs with the bases in the DNA strand. Also, note that the RNA nucleotides contain the sugar ribose, which has a 2' hydroxyl group, whereas the DNA nucleotides contain the sugar deoxyribose, which lacks this hydroxyl group. RNA polymerase functions similarly to DNA polymerase. It adds nucleotides to the 3' hydroxyl group of the last nucleotide. The DNA double helix rewinds as the RNA polymerase moves through. Like the unwinding, the rewinding is an energy-requiring process that is accomplished by RNA polymerase. When RNA polymerase reaches the termination site, the RNA transcript is released from the template. The DNA rewinds completely and the RNA polymerase dissociates from it. The DNA and RNA polymerase can then participate in other rounds of transcription.
Did you enjoy the video? Ladies, boys? Can you all hear me? Yeah, okay, alhamdulillah. So everything was clear about the video. So I have a video for transcription factors as well, but I'm going to upload that on Moodle. I want you to watch a translation first. Okay, give me a minute. I'm trying to upload a translation. Where is it? Give me a minute. Okay. Just let me know if you're not able to watch it or listen to the sound. So here it is.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the amino acyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Yes, boys and girls, how was it? Did you understand anything from it? Clear, isn't it? So now it would be easier for me to make you understand, because transcription translation is something, unless and until you see a video, you cannot get an idea, how actually it is, what exactly happens here. Okay, so let's start with the translation. We just have 7 minutes are left. We just at least finish the charging of tRNA, and then rest of it we can finish later on. Okay, So these are the following steps of the translation. So what happens in translation? What are we doing basically? We are simply forming the protein from the mRNA sequence, right? So what are the requirements of the translation? We need an mRNA, we need a tRNA, we need a ribosome, right? These are the three basic requirements of the translation. Other than that, you will see that we'll also need certain, initiation factors as well, and a small amount of energy as well in the form of GTP. Okay, so what is activation of amino acids? Okay, so activation of amino acids, if you remember, I explained you initially that first of all, we know that protein synthesis takes place from N terminal to the C terminal. We learned that in proteins chapter. Okay, so what is an activation of amino acid? Activation of amino acid is simply, when the amino acid, okay, is activated, okay, by reacting with the molecule of ATP. Okay, so this is an ATP, as you can see here. This is an ATP, okay, and this is an amino acid, right? So it forms as an amino acid AMP, and the phosphate is released, giving us the energy. So this is called as the activation of amino acid. Okay, so how do we activate the amino acid? By simply binding it to the ATP. Now, a second step is the charging of the tRNA. What is the charging of the tRNA? Now, the amino acid which has been activated, that amino acid will go and bind to its particular tRNA, because we said that we have 20 amino acids. We might have 40 to 50 tRNAs, and then we also said that we have a specific enzyme for that. Right? We spoke about that amino acyl tRNA synthetase, if you remember, right? Earlier I explained you about that, right? So what is an activation of amino acid? It's simply that the amino acid will go, as you can see over here, the amino acid will go and bind. So this is the place where amino acid is going to amino acid going to bind the 3' free OH group, hydroxyl group of the tRNA. Okay, and we will have an ester bond over here. Okay, and that is called as the charging of tRNA, and this will take place in presence of an enzyme called as an amino acyl tRNA synthetase. So we just had a tRNA, and now we have a charged tRNA. So we have an amino acid, okay, plus tRNA. Okay, so this is called as the charging of tRNA. Okay, so this arm of the tRNA is called as an acceptor arm. Okay, the arm of the tRNA which binds to the amino acid, that is called as an acceptor RNA. Okay, as you can see here, it's not shown over here. Usually it has three unpaired, not unpaired, it has three free sequences over here. Okay, for example, it might have C, C, and A over here. Okay, so then you will have 3-OH group over here, right? So that is the place where the amino acid will bind. Okay, is it clear till now? So this is the activation of the amino. And if you remember, I told you that this activation is very, very important. I told you earlier, this stage is very important, and the accuracy is very, very important, because it will create a mutation if the right if the right amino acid will not bind to the tRNA. Okay, because after this, there will be no other opportunity for anyone to check that, do we have a right amino acid here or not? Okay, because after that, the anticodon will simply, anticodon of the tRNA will simply match up its correct codon on the mRNA. So whatever amino acid we have here, okay, its tRNA is not bothered. So that is why it's very, very important. This is a crucial step. If there's a mistake in this step, khalas, it can cause a tafra. It can cause a mutation. Okay. Then the second step and the other steps, the initiation, elongation, the termination, these are the other three steps which we will finish in the next class. Ideally, we could have finished this on Wednesday's class, but on Wednesdays, if you remember, I told you I'm going to teach you, I'm going to teach you 2 chapters, which I could not teach you because of when I was infected, right, with the COVID, right? So, which were taught to you by Mr. Hindavi. So I'm going to teach you, 2 chapters on Wednesday. We'll have, back-to-back classes. We'll have a combined class from 9 to 12, even if we have to extend it, it's okay. Okay, so 9 to 12, you will have an extended class, but please remember that join if you have a class at 10:30, you can please leave, okay? So you can listen to the recording later on, okay? So I'm going to teach you the 2 chapters. I might start with the enzymes and the proteins, okay? And then we'll do the neurotransmitters. Maybe we can do that on Thursday, okay? Or on Friday. Would that be okay? So that makes you happy, right? So that will be so that way we can actually cover 2 chapters. Having back-to-back classes, and having two combined classes, will help us to actually finish 2 chapters within 3 hours. Okay, but in between we can take the break as well.